All right, guys, so your final exam is on Wednesday during normal class time. And what I'm going to do is tell you the kinds of things that you probably ought to look over before your final. Because remember, it's 20% of your grades. Yes, I'm going to post this video on Edmodo so you can look at it. I'm looking at a copy of the exam as I'm speaking here. You cannot. There's a couple of questions about basic lab equipment. For example, what you saw me doing just before class started today was to take these little plates we grew the bacteria on and put them in bleach to kill the bacteria. What are those plates? Petri dishes. If we wanted to measure the volume of some kind of liquid, what's the most accurate way of doing that? Graduated cylinder. If we use the little beaker-like containers that have the slender neck on them that you can put a stopper in, what's called Erlenmeyer flasks. There's lots of other things we could use, but those are the three main things that might show up on the test. There's a couple of questions about like your metric prefixes. For example, remember everything in the metric system, SI system, is based on the power of 10. What prefix in the metric system means 10 of something? Deca. Okay. Which one means 100? Hecto. Which one means 1,000? Kilo. And then you go on the other side and you get smaller. Which one means 110? Deci. With an I, deci. All right. 1/100 is centa, and 1/1000 is milli. Yeah.
What's the thing again? King Henry.
It depends on who you hear. Yeah, that works. All right. Another thing that we want to talk about is how experiments are designed. So I'm just going to call that experimental design. In an experiment, remember, you should only have one thing that you're testing. What do we call that thing that we are trying to test? We're consciously changing it to figure out what happens. Yeah, but what's it called? Yeah. What? It's called which variable? The independent variable. For example, if we wanted to know the effect of pH on how much a plant grew, pH would be our independent variable. We'd manipulate that. We might grow some plants with pH 2, and some with pH 4, and some with pH 6, and some with pH 8. That's our independent variable. There should only be one of those in an experiment.
That's the one you're changing, right?
It's the one you're consciously changing and trying to test. Now, because you change the independent variable, you would expect that to change something else in the plant or whatever you're working with, right? What's the thing that you measure that changes because you change the independent variable? The dependent variable. And in the experiment that I just described, what would that be? Now, we change the pH to measure what about the plant? Its height or its growth level. That'd be the dependent variable. All right, something else that's on the test, several questions about, is how things move in plants, or in animals, in cells in general. Cell transport mechanisms. So we'll just say cell transport. The most basic way things move inside cells is called what? The most basic way that substances move inside a cell. What's the word for it? Diffusion. Okay? Diffusion. Diffusion moves materials from high concentration of that material to low concentration of that material. And we say that diffusion is passive because the cell doesn't have to use what? Its own ATP. Okay, it doesn't take a lot of energy. Let's put that away too, man. Pay attention. So diffusion is the most basic way. There's also osmosis, which is the diffusion of what? Water. And remember that water, since it's diffusion, moves from high concentration of water to low concentration of water. But we also can sometimes talk about water moving in terms of the stuff in the water. What do we call things that are dissolved in water? Solutes. So we might say that water moves from where there's less solute concentration to where there's more solute concentration. Or we might throw another word at you and say that they move from hypotonic, which means less, to hypertonic, which means more. That's diffusion and osmosis. There's a couple of other ways things can move, though. One of them is called facilitated diffusion. And that means diffusion with a little what? Facilitated diffusion. Diffusion with a little what? With a little help. Facilitated diffusion are how big things, or maybe ions, charged things, move through a cell. It's still a type of diffusion, so it's still passive. Things are still moving from high to low. And then a fourth one, is active transport. In active transport, things are being moved against what? Against the concentration gradient. They're getting moved from low to high. And that takes what?
ATP.
That takes ATP. That takes energy. And then the final couple ways we'll mention that things move are when you're taking, a cell's taking in big amounts of things. And it can be solids or liquids. If it's solids that they're engulfing, what's that called? When it engulfs something big and solid.
It's
Phagocytosis. Or it can engulf things that are dissolved, and that's called pinocytosis. So that's cell transport. Another thing that might show up on your test are a few questions about enzymes. Enzymes are proteins in your body. What's their job? What do enzymes do? They make chemical reactions happen. Sometimes we say they're catalysts, which means they make things get started. It might be digestion to cause things to break down, but there are also lots of enzymes in your body that make things go together, that build up. Enzymes make reactions happen. They're proteins, so they're coded for by what? DNA. There are several questions about photosynthesis and cell respiration. Remember that photosynthesis takes in what gas? Carbon dioxide, and uses that carbon dioxide along with some water to make what main thing? For the plant, what's the main thing? Sugar, glucose. And oxygen's given off as a waste product. You ought to know for this test that equation for photosynthesis. You ought to know that. And you ought to know the reactants and the products. Remember that cell respiration is sort of the opposite. Remember, in cell respiration, you take in sugar and you break it down and you use the energy in the sugar to make what? ATP. Every living thing does cell respiration. Plant, animal, everything in between. Cell respiration's main job is to make ATP for living things. Cell respiration gives off what gas as a waste product?
Carbon dioxide.
Carbon dioxide, and it needs what gas as a reactant? Oxygen. Oxygen as a reactant, carbon dioxide is given off as a product. The opposite of photosynthesis. You should know, again, the equation for photosynthesis and the equation for cell respiration. All right, a couple of other things on the test are about cells, just cell structure. So what we're talking about first here, most basic thing, like the differences between prokaryotes and eukaryotes. Prokaryotes are the more primitive kind of cell, like what kinds of organisms? Bacteria. Prokaryotic cells are lacking an enclosed nucleus and also what? Organelles. The one organelle-like thing they have is what? Ribosomes. They got to be able to make proteins. On the other hand, eukaryotes have a DNA, that's enclosed by a membrane, a nucleus. They have lots of organelles. For example, they have mitochondria. What happens in the mitochondria? Cell respiration. They have a Golgi apparatus. What does it do?
It stores.
Helps to send things out of the cell. What's called exocytosis. They have the rough ER, which has ribosomes on it and helps to make proteins and then ship them through the cell. They have lysosomes, which help to do what?
Break down.
Break down things inside the cell. Get rid of waste in some cases. You ought to know what those parts do. You ought to be able to look at a diagram of the cell and tell what the parts are. Specifically a eukaryotic cell. There's also some classification questions, which is the stuff we've done recently. You ought to know the order of the different groups, the different taxa. Domain, and then kingdom, and then what? Phylum, and then order, family, genus, species. You ought to know that a scientific name is made up of what? Two parts. Genus and species. The genus name should always be written how? First letter capitalized, species all lowercase, and it ought to be in italics if we're typing it. And everything's going to be typed on the test, so it ought to be in italics. Yeah. Well, sort of. There's some matching too. And then another thing that's on the test is a little bit of stuff about the cell cycle.
What are those genes?
Phylogenetic. Several questions about the cell cycle and stuff like mitosis and meiosis. So we'll say cell division. What's the part of the cell cycle called when the cell's not dividing? Interphase. And remember, interphase is made up of three pieces itself. G1, S, G2. And then one of those you really ought to know is S, because what important thing happens there? Replication. Copying of DNA. Awesome. And remember that the S part of interphase is not part of cell division. Interphase itself is not part of cell division. This is what happens before and after cell division. What's the most basic type of cell division? Mitosis. And remember that mitosis happens all over your body. Ladies, focus. Mitosis happens all over your body. It takes one cell, and you end up with how many? Two. And they are just like the cell you started with. Mitosis is cloning. If the cell started with 46 chromosomes, you get two cells with what? 46 chromosomes. It copies them. It makes identical copies. Its jobs in your body are to repair dead cells, to repair damage, to replace things, to help you grow. All kinds of important things, but all the cells that are made by mitosis are just alike. Remember that mitosis happens in 4 stages. First stage is what? Prophase. Prophase. In prophase, the DNA coils up and makes chromosomes. The nuclear envelope breaks down, and the spindles start to form. What's step 2? Metaphase. And that's where the chromosomes all get pulled to where?
The middle.
The middle. They line up in straight row. And then, what's next? Anaphase. Those X-shaped bivalent chromosomes get pulled apart. One set of chromosomes goes to the right, one to the left. And then telophase, they make it to opposite sides, and essentially all the stuff that happened in prophase gets undone. And then what always happens right after mitosis? Cytokinesis. Remember, mitosis is division of the DNA. Cytokinesis is division of the actual cytoplasm, the cell. Mitosis, ladies, stop what you're doing, pay attention. Mitosis, you start with one cell, you end up with two that are just alike. Now, another kind of cell division that you should be aware of, though, is meiosis. Meiosis happens only in your reproductive organs. It takes one cell and makes 4 cells. Are those 4 cells made in meiosis alike?
No.
They are not. And what do we know about the number of chromosomes in cells made by meiosis? It's half the normal number. So if you started with a cell with 48 chromosomes, you end up with 4 cells, each with 24. Those cells that are made by meiosis become what? Gametes, sex cells. That's the main job of meiosis, to make the sex cells. To make the sperm, to make the eggs. You guys, I'm going to say this one time. If you can't be quiet and focus and pay attention, there will be no review. Okay? You won't do very long in the exam. Whatever else you do, I want you to stop, turn around, watch and listen, look, turn around. The next time I get on to somebody, I stop, I turn off the video, I don't post it. There will be no review, period. All right. So, mitosis and meiosis, know the differences, know what they do, know their functions. There's a few questions about genetics and like Punnett squares. Couple of words first. What does it mean if something's heterozygous? Because one dominant and one recessive allele, different forms of a gene. If it's homozygous, it has 2 that are just alike, right? You ought to be able to do a Punnett square. For example, let's say I told you that you had a parent who was heterozygous, tall. That's heterozygous. And you were crossing it with a parent that was homozygous, short. Short's recessive. So remember, if you want to do a Punnett square for that, meiosis takes these 2 T's and separates them from each other, puts them into different way. What are these 2 T's going to get put into separately from each other? Gametes. All right, these 2 are going to get separated also and put into different gametes. You keep the 2 T's from this parent on the same side and the 2 from this parent on a different side. You cross them. Fill in the boxes using a grid pattern. We call these the genotypes, right? The symbols. What do we call what they look like? The phenotypes. So we have 2 tall phenotypes up at the top, and we have 2 recessive phenotypes down at the bottom. 2 talls, 2 shorts. Yeah.
Like that, then like you put like a C at the top.
Yeah, you're not gonna have anything like that for right now. Another thing that's on the test is some stuff about how DNA works, the DNA structure and function. First of all, DNA is a nucleic acid. What are nucleic acids built out of? What are their monomers? Nope, that's for protein. What are the small pieces that make up DNA? What are the monomers though for DNA? It's made of a sugar connected to a phosphate connected to a nitrogenous base. Those are called nucleotides. Nucleotides. And DNA is made of 2 chains of those. 2 long chains of nucleotides. Therefore, it's called a double what? Helix, because they get twisted. We built a model of that. You ought to be familiar with that. What holds the 2 chains together in the middle?
Weak hydrogen bonds.
Weak hydrogen bonds between what? What part of the nucleotides connect in the middle?
The sugar.
Nope.
No.
A and T and G and C. Those are the nitrogenous bases, right? So the 2 strands are held together by bonds between the nitrogenous bases. Remember that T always has to match up with A and G matches up with C. Something else that'll be on the test are a few questions about what we call the central dogma of genetics. Remember, the central dogma is how the genes work. How the genes ultimately make what? What are genes trying to make? Proteins. So, let's think about that central dogma for a second. We know that the genetics start with DNA, and that the DNA can be used to make RNA, and I hear the phone, but I'm ignoring it. That process of taking the DNA and using it as a template to make RNA, what's that called? This is called transcription. The mRNA, focus please. The mRNA will then leave the nucleus and it'll go out to the ribosomes where it's used to make what? mRNA is the directions for making what? Proteins. And we call that process what? Yeah, or what's the T word for it? This is transcription right here. This is translation. And then those proteins will go on to make your traits, to determine your traits. That's what we call the central dogma of genetics. Right now, just listen. All right, there's also some questions about the stuff we just finished doing. Food webs, energy pyramids. In an energy or trophic pyramids, what's the bottom level? Producers. What are the things above that? What kind of consumers? Primary. Things that eat the primary consumers are secondary, then tertiary, then quaternary. And slow down. And remember that each level you go up, only about 10% of the energy is transmitted up the chain. So the further up you go up the chain, the less and less energy is available. And therefore, there's less and less organisms up there as well. So it's called the rule of 10. We also talked about food webs. And we know that in a food web, the arrows always point toward what? What's doing the eating. Okay, the eater. The arrows point toward the consumer. Another thing that we talked quite a bit about the last few days was symbiosis. Remember, symbiotic relationships are relationships where two or more organisms are either working with each other or against each other. We talked about mutualism, where they're helping each other. It's a good deal for both. We talked about parasitism, where one thing benefits in the relationship and the other's hurt, like a tick in a human. And we talked about commensalism. Commensalism is where one benefits and the other one isn't helped or hurt. We talked about the little remores that live on the sharks. They don't hurt the sharks, they don't help the sharks. But they feed on like the scraps after a shark eats.
So like a mosquito here?
Mosquito would be parasitism, right?
So that's
Mutualism and commensalism. There's a few questions about something else we've done recently, which are the cycles, the biogeochemical cycles. The carbon cycle is mostly controlled by what two processes? Carbon cycle. Photosynthesis takes carbon dioxide out of the air, makes sugars, and cell respiration takes those sugars, gets energy out, and puts CO2 back into the air. It's the carbon cycle. The nitrogen cycle, on the other hand, where's most of nitrogen on Earth?
In the water.
It's in
It's in the air. In a form that most things can't use. So some bacteria can take that nitrogen out of the air and do a process called nitrogen fixation and turn it into usable forms. And there are other bacteria in the soil that do the opposite. And that's called denitrification. And it just keeps cycling it back and back and back and back and back. All right, there's also several questions about viruses and bacteria. What's the virus reproduction cycle called?
The lytic.
The lytic cycle, where they attach to a cell, inject their DNA, reproduce, and then kill the cell. Remember that viruses have to live inside other cells, so they're called obligate what? Intracellular parasites. And remember that virus diseases can or cannot be treated with antibiotics. Cannot be. Antibiotics are for bacterial diseases.
Are we going to get our test back?
Right now, The different shapes of bacteria. Remember, they come in a coccus, which is like a ball shape, a sphere. They come in a bacillus, which is like a rod or a capsule, and a spirillum, which is kind of like a corkscrew kind of shape. Remember that bacterial diseases are treatable with antibiotics. And there's lots of bacterial diseases, everything from the bubonic plague to tuberculosis to tetanus to staph infections. A lot of STDs are bacterial. And then finally, the last thing that's going to be on your final are several things that are going to be on this test that I'm talking about for Monday. There's going to be a diagram of the frog, which you see right here. Some questions about that diagram. Labeling kinds of questions or function kinds of questions. Your midterm, or your final rather, is 80 questions. And it is all either multiple choice or the diagram part is matching. That's what it's going to look like. If you know the things I just talked about on the video, you're going to do really well. Remember that this thing is 20% of your grade. So it's important, since it's 20% of your grade, for you to look at it. Since there's 80 questions, each question is like 1.2, 1.3 points apiece, something like that.
It's the first first 9 weeks times 0.4, second 9 weeks times 0.4.
Right, the way your grade works, the first 9 weeks grade is 40%, second 9 weeks grade is 40%, and the final exam is 20%. So this is more than just a test. It's a very important test. You need to take some time and study. We're not going to have a lot of review time in class. This is most of the review time we're going to have. Because on Monday, we're going to take the test on the frogs. We're going to start preparing for the dissection. On Tuesday, pretty much the whole class is going to be dissecting the frogs. Test is on Wednesday. Ain't for mine. All right, so let me stop the video, and then I will answer some questions for you guys. Cut for a minute.